Hello students, welcome back. In today's lecture, we will discuss how professional antigen-presenting cells present antigens to the immune system using the MHC. Remember from a previous lecture that non-professional antigen-presenting cells, APCs, can only present antigens once they have been infected. Let's draw our professional APC, antigen-presenting cell. Here we have a cell with its nucleus. On this cell, we have a very special molecule which binds very specifically to microbes. Traditionally, this molecule is represented with a Y shape. Our next element is a new type of MHC which has two legs rather than one. This new element is MHC2. Remember that the MHC1 has only one leg going through the membrane. The next item that we will need is a hydrolytic vesicle which contains enzymes to hydrolyze the microorganism. Our final component will be the microbe itself. We could use either a virus or a bacterium, but I've chosen to use bacteria. As the bacterium floats through the body, it will be bound by the Y molecule. Next, the cell will phagocytize the bacterium. Let's redraw our cell in the process of eating the bacterium. Here we see the cell in the process of phagocytosis. Also, we see that the hydrolytic vesicle starts to fuse with the phagosome. Let's show the completed phagocytotic process. Here we see that the bacterium has been completely digested into fragments. These fragments are bound to the MHC2 molecule. In the next step, the phagosome then refuses with the membrane. Here, the Y molecule and the MHC2 are on the surface. We see that the MHC2 molecule is presenting its antigen. A special immune cell recognizes this antigen. Let's draw this new cell. This cell is the master cell of the specific immune system. It has a receptor which binds to the antigen, and it also has a molecule which binds to the MHC2. Once the special immune cell binds to the MHC2 molecule, it releases chemicals to activate the professional antigen-presenting cell to fight the pathogen. We will show this activation process in the next lecture. Let's annotate the items in this lecture. Here we have a bacterium, the nucleus, a hydrolytic vesicle, and the MHC2 molecule. The one new item that we have is the Y-shaped molecule. This molecule is called an antibody. And these are all of the items in step 1. On the second cell, we have phagocytosis of the bacterium. Phagocytosis is cell eating. The Greek word phago means eat. And this is all we'll annotate here. On the third cell, we have bacterial fragments, the antigen, and the phagosome. Because these fragments come from a bacterium, they would all be antigens. Since the cell here presents an antigen on the MHC2 molecule, we call it a professional antigen-presenting cell, abbreviated APC. This cell is a B lymphocyte, or a B cell. The antibody is also called the B cell receptor. And that is all we will annotate here. The cell which recognizes the MHC2 molecule is the master cell for specific immunity. This cell activates B cells, cytotoxic T cells, and macrophages. The chemicals which are released to activate the other cells are called cytokines. This master cell is a helper T lymphocyte, or a T helper cell, or a helper T cell. It is abbreviated T subscript H. The molecule which binds to the antigen is the T cell receptor, TCR. The molecule which recognizes the MHC2 is the CD4 protein. CD4 proteins are primarily found on helper T cells. For this reason, the helper T cells are also called CD4 plus cells. One interesting fact in this lecture is that one of the primary targets for the human immunodeficiency virus, HIV, is the T helper cell. In the next lecture, we'll see how the B cell is activated to perform its primary function. So that concludes this lecture on professional antigen presentation using the MHC2 molecule. I hope that you've learned a lot.